Hello freshmen, welcome. More on viruses. Highly topical issue here. Notice what we have is three different viruses. We've already talked about the bacterial phage here, or bacterial phage, and we talked about the parts. Now this is a DNA virus, and notice we have an electron microscope picture of a bacterial phage that has attached to a bacterial membrane and is about ready to inject its genetic material into that bacteria. Notice the size there. That is 60,000 times magnification. Again, these viruses are specific. They attack specific cells which have specific receptors. Notice here we have the flu virus. Here's our capsid. We also have RNA as the genetic material. We have surface proteins, and I do want you to make a picture here. So we're going to write flu virus, and let's draw a picture. Notice the electron microscope picture. Make sure you label the surface proteins, the RNA, the capsid protein, and then we also have this membrane envelope. We'll see with the HIV virus that it also has some similar structures to this. The coronavirus is related to this as well. So the coronavirus is an RNA virus. These surface proteins are important because they allow an attachment. You almost have a lock and key sort of fit where the virus has these surface proteins which are acting as the key, and then the respiratory cells, okay, so respiratory cells, whether it's the flu virus or whether it's the coronavirus, has specific receptors on the membrane which these proteins here can attach to. Again, a lock and key sort of fit. Now, one big important difference here in terms of the flu virus versus the coronavirus, typically the flu infects, and this is going to be a lytic virus, and you're going to feel sick pretty quickly after you've been infected. Of course, hopefully after maybe 4, 5, 6 days, your immune system will ramp up a defense and then start deactivating those viruses. So the coronavirus has more of a time lag after infection. So you might go 10 days before you're feeling sick. So let me get rid of this, and let me get rid of this. We are going to take a look. Oh, I'm sorry. Let's also take a look. There's the tobacco mosaic virus. So this infects bacterial cells. And again, this is fairly simple. We've got the capsid protein. I do want you to draw all three of these. You already have this actually, so let's draw these and label. Again, we've got an RNA virus here, and there is the electron microscope picture. Viruses tend to be very specific, and the polio virus will attach to nerve cells in the spinal cord and infect their genetic material, and then we're going to get a lytic cycle infection. Well, what I want to do now is look at the HIV virus. So this causes the syndrome known as AIDS, acquired immune deficiency syndrome. This HIV virus, again, is responsible for about 35 million deaths worldwide since the outbreak in the early 1980s. This is also a good example of a lysogenic virus, and it's also a good reminder of some important aspects of protein synthesis as well. We're going to use this HIV model kind of as a way to help us understand how retroviruses, and we'll talk about what a retrovirus is here in a moment, how retroviruses work, infect cells, reproduce, and wreak havoc within the human body. So let's get to that. So here is the HIV virus. We have these specific proteins here on the surface. We then have a envelope which surrounds the virus, and then a protein capsid is going to be on the inside. Let's watch the video and then I'll stop it, and I want you to draw a picture of this and label all the parts to this virus. Please don't call this a cell. It's not a cell, okay? This is a viral particle. It's non-living at this point. Okay, so you're going to draw a picture here, and the first thing you're going to label here on the inside is going to be this RNA. Remember, this is an RNA virus, and there's 2 strands of RNA. So go ahead and get that.
Enzyme here in blue that's reverse transcriptase.
This virus brings with it its own special tools that it's going to need to take over the cell once it infects the cell and enters the cell. Reverse transcriptase, we'll take a look at what that does, but for right now, think of it as a tool that the host cell doesn't have. So this virus here has to bring this special tool with it. So what's the role of reverse transcriptase? Remember, this is RNA, so reverse transcriptase is an enzyme, and you need to get this, is an enzyme that reverse transcribes this RNA. So in other words, it takes the RNA that the virus has, and it's going to perform reverse transcription and produce DNA. It won't be able to do this until it enters the cell, though. So we have RNA, we have reverse transcriptase, which converts viral RNA to viral DNA. Go ahead and make a note. We have I'm not sure if they're going to mention it, but these are special protein-like structures. I want you to get the function of these special protein-like functions on the surface of the virus. These are the keys that unlock the immune cell that's going to be attacked here, and that's what I want you to get. So this provides the key, quote unquote, that's going to unlock the specific receptor of the immune cell in our blood. Let's see what else we got, what else we have.
Which will ultimately turn that RNA into DNA. The virus itself, with this outer envelope protein, actually directly infects T helper cells. The way that it does this is that as it comes up to the cell surface, it uses receptors that are on T helper cells and exclusive to T helper cells, which are CD4 molecule, which really defines T helper cells. It's a surface receptor that binds to the envelope protein.
So this is the immune cell. And it has a special name. It's a T helper cell. But think of it as an immune cell. It's going to help, normally it's going to help fight disease. Now here's the specific receptor on the immune cell. It's called a CD4 receptor. This is the protein-like structure that's on the HIV virus. And here is the outer envelope with the protein structure of the HIV. So here's the HIV. And this is the surface of the immune cell, of the T helper cell.
That causes a conformational change and allows a second receptor to grab hold of the envelope. This is the chemokine co-receptor. It's also called CCR5, and we'll talk about that more. What happens now is that the stalk of the envelope protein pierces through from the virus into the host cell and starts to draw the cell membrane and the viral membrane together. And what ultimately happens is fusion of those two membranes, and the viral genetic material is injected essentially into the cell, and the envelope protein is left at the cell surface. The virus has a matrix and a capsid protein, shown here in green and red, that essentially are digested when it enters into the cell. That releases the viral enzymes and the viral RNA. And here we have reverse transcriptase, which takes the viral RNA and, using host nucleotides, converts that viral RNA into a single strand of DNA. While it does that, it makes...
So look at what's going on here. So this is reverse transcriptase. This is viral RNA. Notice reverse transcriptase is converting this viral RNA into DNA. So this is viral DNA. Notice, where do the nucleotides come from so that this virus can convert its RNA into viral DNA? It comes from the host cell. So again, this idea of commandeering or taking over the host cell and using all the components of the host cell to ultimately make more viral particles, ultimately. So typically with DNA, we're going to express our genes by converting the DNA into RNA, so that's transcription, and then the RNA is converted into a protein, so that's translation, and we've talked quite a bit about that. This is interesting and important. This is what a retrovirus does. A retrovirus is going to have reverse transcriptase, and it's going to convert the viral RNA back into the DNA. Let's see what happens next.
Some random errors, which is characteristic of reverse transcriptase. It has very poor proofreading activity. That single-stranded DNA now is again reverse transcribed into a double-stranded DNA. At that point, another enzyme that has come in with the virus in the beginning, called integrase.
Okay, notice, go ahead and write integrase. Integrase has come in with the virus. So this is another protein that's in the viral tool chest. So go ahead and write integrase. Again, make a note, this came in with the virus in the tool chest. Now look at what integrase is going to do.
Essentially grabs hold of that double-stranded DNA and carries it through a nuclear pore into the nucleus of the cell. Within the nucleus of the cell, it finds the host chromosome, and it basically, the integrase enzyme, makes a nick in the host DNA and allows for HIV to insert itself into the host chromosome. And that right there is what establishes lifelong infection.
All right. What we have now is viral genes that has incorporated itself into the host set of chromosomes here. This now is called a prophage. So remember, a prophage is the inactive form of the viral DNA. And notice, what better place to hide from the immune cells than in the DNA of an immune cell. So this is an insidious, there's a good word there, an insidious HIV viral DNA that is, it's like a needle in a haystack, just this tiny prophage in this gigantic amount of DNA of the host cell. And right now, it's inactive. But like all lysogenic viruses, it will become active at some point in the future. Again, HIV might become active after infection 5, 6, 7, 8 to 10 years after the initial infection. Well, let's see what happens now when this prophase, when the HIV virus activates.
Now, RNA polymerase comes along and makes messenger RNA. Those messenger RNAs encode for different viral proteins. They end up associating with ribosomes at the surface of the rough endoplasmic reticulum. And here's a piece of mRNA that's making envelope protein, which is directly produced into the endoplasmic reticulum. And it's shuttled then through the endoplasmic reticulum and taken to the cell surface, where at the cell surface it becomes embedded in the cellular membrane, and at this point coalescing with other envelope proteins that have been produced. You have this cluster of envelope proteins now on the surface of this infected cell. At the same time, there are other messenger RNAs that are being produced that allow for translation of other viral proteins. So here are additional viral proteins being made, which are going to be used to make up the key components that the virus ultimately is going to need. These are transported again to the cell surface, to the area where these envelope proteins are, and a strand of RNA as well as some of the enzymes are part of that complex. This then buds off at the cell surface at this point, but it's still not a mature virion because the polyprotein chain needs to still be digested into its component parts. That's done by an enzyme called protease. Protease breaks up those polyprotein chains and ultimately allows for them to coalesce and form the mature structures that make up the final virion. And now you have a mature...
So go ahead and write protease. Protease. Protease was responsible for creating a mature HIV virus, which can now go out and infect other cells. One more time, protease. It's responsible for creating a mature HIV virus, which now can go out and infect other cells. Also, boy, that was just a great sort of visual review of what happens during protein synthesis, where the DNA is converted to messenger RNA by transcription. Then the, class is not over. Class has just started. This virus and bacteria stuff, we're about halfway through, so we've got more to do. It's all not going to be on this video. But let me get back to what I was saying. That's a great visual representation of DNA being converted to mRNA. In fact, let's go back and re-look at that.
Grabs hold of that double-stranded DNA and carries it through a nuclear pore into the nucleus of the cell. Within the nucleus of the cell, it finds the host chromosome, and it basically, the integrase enzyme...
There's integrase, an enzyme.
DNA, and allows for
there's a prophase
HIV to insert itself into the host chromosome. And that right there is what establishes lifelong infection. Now, RNA polymerase comes along and makes messenger RNA.
Transcription there.
Those messenger RNAs encode for different viral proteins. They end up associating with ribosomes.
This is translation out with the out in the cytoplasm. There's the ribosome.
It's making envelope protein, which is directly produced into the endoplasmic reticulum, and it's shuttled then through the endoplasmic reticulum and taken to the cell surface, where at the cell surface it becomes embedded in the cellular membrane. And at this point, coalescing with other envelope proteins that have been produced, you have this cluster of envelope proteins now on the surface of this infected cell. At the same time, there are other messenger RNAs that are being produced that allow for translation of other viral proteins. So here are additional viral proteins.
More translation. There's the protein, viral protein.
Make up the key components that, that the virus ultimately is going to need. These are transported again to the cell surface, to the area where these envelope proteins are, and a strand of RNA as well as some of the enzymes are part of that complex. This then buds off at the cell surface at this point, but it's still not a mature virion because the polyprotein chain needs to still be digested into its component parts. That's done by an enzyme called protease. Protease breaks up those polyprotein chains and ultimately allows for them to coalesce and form the mature
Again, that's the action of protease.
Make up the final virion. And now you have a mature infectious virion that can go on now to infect other cells. Once that happens, now the cell can produce tons of viruses, and this is really what then keeps the whole process going.
So that immune cell, that T lymphocyte, has become a virus factory. It's releasing huge number of viruses. Eventually that T cell is going to die, and all those viral offspring are going to go and infect new healthy T4 lymphocytes, immune cells, and have the same effect. Over time, once you reach the lytic stage, so that was the lytic stage there, once that point is reached, the immune cell numbers plummet. They go way, way down. Eventually, if this is untreated, the immune system is so compromised. AIDS is acquired immune deficiency syndrome. So once they go into full-blown AIDS, where you have the lytic cycle and you have this precipitous drop in immune cells, typically an AIDS patient is going to die of opportunistic infectious particles. These would be maybe bacterial infections that you and I with healthy immune systems can easily fight off. But these people, these afflicted patients cannot fight off typical bacterial infections. So they'll end up with a certain type of pneumonia. Again, it's really, you can kind of see why we spend so much time talking about DNA, RNA, and protein synthesis, because it directly relates to this sort of process, which is infectious disease. All right. We're going to be talking more about viruses, and we're actually going to give the good stuff about viruses, and that's going to be on the next video. After that, we're going to go to bacteria. Have a good day.